Thank you, Donald, for a lovely invitation and the introduction and the organizers. It was definitely worth the effort. I really enjoyed the talks we've had so far, and it's absolutely beautiful, my first time in Lausanne, so thank you for having me. So yeah, I've got a lovely picture up there of our campus. It's a beautiful campus, but unfortunately, that's not where I'm based. We are based at the hospital. And the reason we've been there is, so immune aging is, of course, an area of key interest, but we also work with a number of other cohorts which show accelerated immune aging. So we're actually based in the hospital to get access to a lot of these cohorts. So why are we really interested in aging? Personally, from my point of view, if we look at the demographics here, so this is from a House of Lords report from the UK. What's really interesting is, of course, our lifespan's increasing. But what I want to draw your attention towards is the two lines at the bottom there. Over here. So although our lifespan is increasing, what hasn't kept pace with this increased lifespan is the health span. So although we are living longer, on the average, older adults spend about 7 to 11 years of their lives at the end in poor health. And this is what we're aiming to target. So what we want is not to possibly increase the lifespan, but decrease this period of age-related diseases and promote healthy aging. So what's causing this big gap between lifespan and health span? Firstly, we've heard a lot about this. We start seeing increased risk of infections, whether it's bacterial infections, viral infections, so I'm not going to dwell into that a lot more. But it's not only an increased risk of infections. We also see poor outcomes due to this infection. And the reason I've got this study here is that it highlights this really well. So this is looking at your influenza instances. What we have in blue here is the winter periods. The green line here starts showing when you go to the primary care admissions to your GPs during these winter periods. What we can see here in a 45 to 60-year-old cohort, during the winter season, we start seeing these episodes of GP visits due to influenza. But once we progress to the older demographics, this is from our 65 to 74-year-olds, what you can start see emerging is this blue line over here. And this is showing the number of hospital admissions now due to this influenza. And if we further move down to our 75-year-olds, we also start seeing the yellow line, which is the deaths due to these infections. So it's not only that we see an increased risk of infections, it's also we see poor outcomes due to these infections. And of course, the key strategy that we have to combat this would be vaccinations. But we've already heard that with advancing age, we see an impaired ability to mount a robust vaccine response. And this is the case for whether it's influenza vaccines, and this is a lovely example shown over here, where you see a decline in your fold increase of your antibody titers in older adults. But it's a similar case for pneumococcal vaccines or tetanus vaccines as well. So not only do we see an increased risk of infections, also certain autoimmune diseases. So this is an example of rheumatoid arthritis. Some epidemiological data here, you can see that the peak of incidence starts occurring towards later life. And the reason for giving all of these examples above was that as an immunologist, I would like to believe that your aging immune system is one of the key reasons why we start seeing this big gap between our lifespan and our health span. And by targeting our immune system, that's how we can possibly bridge this gap. So I've already discussed about how immunosenescence, which we've heard a lot about, is our remodeling of the immune system, and the key take-home message is that it's an impaired ability to mount a robust immune response. This could be contributing towards increased infections, autoimmunity, poor vaccine responses. But what's really interesting, and there's new evidence that came out in the past 2 years, and what this is showing using animal models is that this immunosenescence could actually be a driver of biological aging. So using animal models, if we age the immune system of a young mouse, you can actually trigger other effects, aging-like effects in other systems. For example, it could induce musculoskeletal aging or other body system agings as well. So it's a really important target to promote healthy aging. So Donald has introduced us to the thymic atrophy and spoken a lot about it, so I'm not going to spend a lot of time dwelling on this, but just to remind you all, some of the key features is we start seeing this shrinkage of thymus, and we've already discussed in the talks earlier on about how we see the shrinkage of your naive T cell pool, expansion of the memory pool, although the overall T cell profile remains the same. But what I want to talk a little bit more about is the other hallmarks of T cell aging. Particularly an area of interest is these accumulation of senescent cells, which we've heard a little bit about already. So these are those cells that display proliferation defects, for example. So to give you an example, what you might think is that, okay, we don't have enough naive T cells, so our ability in old age to maybe combat new infections, such as your SARS-CoV-2, is impaired. But at least we have the memory T cells. So if we re-encounter an infection that we have previously seen, we'll still be okay. But unfortunately, that's not the case. And that's why we at times start seeing emergence of previously infected infections. So one very good example for this one is your VZV virus. We get chickenpox when we're little, but shingles starts reappearing in older adults. The reason is you might still have these memory T cells pool, but that doesn't mean they're very functional. A lot of them have lost their ability to expand when they re-encounter an infection. This is due to the buildup of these senescent cells. And these senescent cells, they show a lot of the key hallmarks of aging. One of their key features is that they're pro-inflammatory in nature. They have this secretory phenotype. They churn out a lot of pro-inflammatory cytokines. They show defects, a buildup of DNA damage. They have defects in their DNA damage repair mechanisms, mitochondrial buildup, defects in mitochondria, autophagy. So they show all the key hallmarks of aging. The other area of interest particularly is these changes in our CD4 helper differentiations. So we start seeing an accumulation of these Th17 cells, which are known to contribute towards autoimmunity. And on the other hand, we have these regulatory immune cells. So these are those subset within our T cells that are anti-inflammatory. They can also help control inflammation. And what's interesting is that we've also seen an expansion of this subset as we grow older. And there's now emerging evidence suggesting a link between expansion of these Tregs and senescent T cells. So they could possibly be a compensatory mechanism. So we've heard a lot about inflammageing as well. And the senescent T cells I was talking about earlier on is one potential contributor towards this, where this universal hallmark of aging, where we see a consistent basal increase in pro-inflammatory cytokines. And the reason why this is not particularly good news is that this could drive systemic effects and contribute towards other features of aging as well. So there's a lot of evidence linking this inflammaging with mortality, with a decline in cognitive functions, with development of sarcopenia or frailty. So targeting this is really important. So hopefully I've convinced you all now, and with the talks we have heard earlier on, that targeting our immune system and having a healthy immune system is the key towards successful aging. The two key ways we could possibly do this. So of course we have our pharmacological interventions, but what I want to talk to you about today is possibly there are a lot of lifestyle-based interventions or modifications that could have really promote healthy immune aging. So in the past, we've done some work on the benefits of exercise on thymic aging. But what I'm going to talk to you about is recent work that we've been doing on manipulating the microbiome and how we could use this to promote immune aging. So why the microbiome? We have heard a little bit about intestinal barrier dysfunction. So Caroline mentioned that. And what I want to draw your attention towards is that, so for many years, we know that there were two things that were occurring in parallel. We start seeing immune senescence, your immune system is changing, and in parallel, we start seeing changes in your microbiome composition. And only emerging evidence now is linking that there are actually not two changes that are occurring independent of one another, and there's a lot of crosstalk between these two. So as we get older, we start seeing changes in our microbiome composition profile. So just to summarize it very briefly, we start seeing a loss of these commensal beneficial bacteria. We start seeing an expansion of the opportunistic ones. And we also see, in addition to changes in the profile, we start seeing changes in the metabolites that they are producing as well. So if you see a loss, for example, of your commensals that are very important in producing your short-chain fatty acids, which we've heard about as well, this is driving this loss of short-chain fatty acids levels both in your gut, whereas in circulation as well. The reason why these are really important is you might think that how can your microbiome sitting in the gut actually impact systemic immunity? And one of the key ways it does this is via these signaling molecules. And these changes in the microbiome profile are a key contributor towards this intestinal barrier integrity, which is lost. And what we start seeing is this translocation of microbial products into circulation. And the only evidence we have so far is of linking these two processes has shown that there's actually a causal relationship between this microbial translocation and inflammageing, which was showed in an animal model of aged germ-free mice. And this is something we were very fascinating in finding out a little bit more about. So going back to the simplistic thinking, if we go back centuries ago where the doctors weren't sure about the differences between different diseases, what's causing them, what are the underlying mechanisms, they were all saying that it all arose from imbalances in your stomach. And we wanted to go back, take a step back and think that could it be that these changes in your gut physiology could actually be driving immune aging? And what is the relationship between these two? So to do this, we first did this study in humans. We recruited 40 healthy young people and 55 healthy older adults. So we had a very strict inclusion criteria. So they were all absolutely healthy, no underlying comorbidities, no infections, no hospital admissions in the past 6 months. A really strict one. This is a very, very healthy cohort that we had. And what we did was we collected blood samples for them, and the first thing we wanted to look for was these circulating markers of intestinal barrier dysfunction. And what we found was that in our older, so it's no surprise we start seeing increased gut leakage in humans. But what was really interesting is the spread of the data that we see here. So Caroline already mentioned about the heterogeneity that we see. And what you can clearly see here is that there are two cohorts within this older adult population who are showing the gut leakage levels just as low as young individuals, but they are the small cohort up here which are showing very high levels of microbial translocation and gut leakage. And what we wanted to test was that if we split our older adults cohort into those showing low levels of gut leakage versus those with high, would we actually see any differences in their immune aging profiles? So to do this, we have done a lot of flow cytometry looking at a number of different T-cell immune parameters, some of which you've already heard about. So the first one, looking at our naive T cells, which are really important antigen inexperienced cells for combating infections. What you can see here is, of course, you see a decline, fewer number of these naive CD8s in our older cohort, but there's a very strong association between the circulating levels of occludin, which is one of your gut surrogate markers for your intestinal leakage, and the levels of these naive CD8 T cells. Then we've also looked at the recent thymic emigrants. So these are those antigenically naive T cells that we've heard about earlier on that have just completed the differentiation and left the thymus and come into the periphery. And if we've known that the levels of these decline with age, but what was particularly interesting is that if we split these older adults into those with low levels of gut leakage versus those with high, we only saw a significant decline in these circulating recent thymic emigrants in this cohort that was showing high levels of gut leakage. So this was the first evidence of hints that this barrier dysfunction could actually be impacting our thymus possibly and the output. So what about the other features of T-cell aging? So we've gone through this earlier on, so I won't spend too much time on it, but how our naive T cells in response to infection get primed, they can differentiate into effector T cells. And what we start seeing with age is a buildup of these T cells with a senescent and exhausted phenotype. And when we look at these, again, it's been known for a while that you see an accumulation of these CD28 negative, CD57 positive T cells with age. But what we found was that there were no differences between the young and those older adults who had low microbial translocation. This buildup of senescent T cells only occurred in individuals who were showing high gut leakage. So I mentioned earlier on about these links between your senescent T cells and regulatory T cells. So when we measured our regulatory T cell profile as well, a very similar trend where we were seeing more of an accumulation of these Tregs in those older adults with high microbial translocation. So we have discussed about a number of different immune biomarkers: naive T cells, memory T cells, senescent T cells, regulatory T cells. And the one key thing we keep having discussions about is which one would be the best or the most accurate marker to predict on what sort of an aging trajectory you're on. So to address this, what we've tried and do is, and this is with support from our collaborators who are bioinformaticians, is that we can actually combine a lot of these immune features to generate one score. So what this is it's called your immune age score. It's a metric in which we can combine 24 different immunological markers to give you one score between 0 and 1. And this is a clinically validated biomarker in which, in a study, they showed that based on your immune age, you could actually predict mortality 6 years down the line. And what we have done now, so this study that was published back in 2019 was relying on 24 immune parameters, and we have narrowed this down now to 12. So we can combine all the different parameters I've mentioned about and generate one score. So we wanted to see whether your immune age score linked with the levels of gut leakage that we had. And what we found here was, looking at two different markers, there was a significant association between the levels of gut leakage and this immune age score in our older adults. So this is providing compelling evidence of strong links between changes in your gut physiology and systemic immune aging. But some of the key challenges with human studies are that they're all associations, and it's very difficult to establish a causal relationship between these two. And to do so, what we have done then is, the other thing we were very interested in was possibly understanding the molecular mechanisms on what's driving these changes or what the differences are. So we've also done some gene expression analysis here. So we have looked at the key hallmarks of aging. So these are those features that are universal, irrespective of which system we're looking for, whether it's the immune system, skeletal system, musculoskeletal system, brain. So these are some of the key hallmarks of immune aging we looked for. So for example, markers for cellular senescence, the ability to clear apoptotic cells, buildup of DNA damage. And what we've done here is we've compared the gene expression profiles between our young cohort and our old cohorts with low microbial translocation and high. And what we found here was, so your autophagy process is very important in clearance of damage, and we only start seeing defects in these in our older adults with high microbial translocation. Similarly, buildup of these senescence markers was also only seen in this cohort. So confirming that they do start seeing key hallmarks of aging. So far, and that's why I go back to the inclusion criteria that we mentioned about, we were looking at this population that was very active, very healthy, but we don't think this is a true representative populations of the actual older adults out there. They're not this healthy, they have underlying comorbidities, they have other things going on. So what we're moving towards now is possibly understanding about how is this dynamics, or how are these interactions between your gut physiology and immune changed in those individuals who are possibly not on a healthy aging trajectory. So to do so, what we have done is we have recruited patients who have just recovered from a hip fracture surgery. So they've had a very complicated surgery, and this is 6 weeks down the line. So whatever effects we are seeing are not possibly a result of the actual surgery. And what we saw here was that with age, we do see an increase in your microbial translocation, but this was significantly higher in these frail older adults. And when we are trying to dissect these interactions with the immune system, again, you see a further loss of these naive T cells, for example. This is even more accelerated in these frail older adults. So this additive effect of a stress, or those individuals who are now, we are working with individuals with multimorbidity who have more than one underlying conditions. And these effects are even more staggering in such cohorts. So Going back to the establishment of a causal relationship between these two, as I said earlier on, with humans, the key drawback is this is all associative data. So to actually see what was going on, we have used a mouse model of germ-free mice. And the reason for choosing these are that they are protected from this age-related intestinal permeability. So this is showing some imaging of the occludent staining between an old wild type, so this is a 22-month-old mouse, and germ-free, who show maintained intestinal barrier integrity. And the published evidence has shown that they are also, as I mentioned earlier on, protected from inflammation. So these aged mice do not show elevated levels of TNF. And what's even more interesting is that if we transfer the microbiome, so germ-free mice are those that are raised in very unusual conditions, they don't have any microbiome. But if we transfer, for example, if an aged germ-free mice receives the microbiome components of a young mouse, you start seeing this increased levels of TNF. But this is even higher if they're receiving components of the aged microbiome mice. So again, hinting towards a causal relationship between microbiome transfer, loss of this intestinal permeability, and inflammageing. But what we were particularly interested in is seeing what happens to our thymus of these mice. So we have heard from Donald's talk earlier on that with age, we start seeing a disorganization, loss of the thymic microenvironment. And that's one of the first things we looked at. So what we've got here is, this is our young type, wild type mice. We are looking at staining for lamin. So lamin loss is considered as a marker for senescence. It plays a very important role in your thymic epithelial cell structure. So with age, you can see clear differences in the organization. The other thing that starts emerging here is you can see these little black voids, which I'll come back to later on. So this is your young mouse aged, and this is our aged germ-free mice that is protected from intestinal barrier dysfunction. And what you can clearly see here is the difference between the microarchitecture. You have fewer cells that have lost the lamin expression, fewer senescent cells here. The other thing is we start, another key feature of aging within your thymus is we start seeing losses of cells within these medulla environments. There are a lot of CD8 T cells, for example, here. Again, if we look at our aged germ-free mice, we see maintained expression of ERT5 here, which is a marker for epithelial cells in these medullary area. So again, showing overall that they have a maintained architecture. Now moving on to these voids that we were talking about, there is a lot of evidence that with age, we start seeing an infiltration of adipocytes, which could possibly be those black voids that we were seeing. And when we did some oil red staining, what we have seen is that, as expected, in this aged wild-type mice, you have a lot of these lipid droplets, which aren't seen in our old germ-free mice. So they're definitely showing maintained thymic architecture. So hopefully by now, I've convinced you about these very interesting links between changes in our gut physiology and thymic aging and T-cell aging. But why this might be of interest is, and the reason of the whole interest in dissecting these interactions was that this is modifiable. Our microbiome is modifiable. By modifying our microbiome composition, we can have a beneficial effect on our gut physiology. So the first possible evidence that came for this, and that's why we got interested, was via fecal microbiota transfer. So I showed you in the aged germ-free mice that if we transfer components of a young microbiome versus an old one, the aged germ-free mice receiving the old microbiome composition start showing inflammageing. So that was one of the evidence, but there's very limited evidence in humans. There's some of the evidence that we do have of a beneficial effect of FMT actually comes from intestinal barrier dysfunction patients. So the fecal microbiota transfer involves transfer of healthy microbiome into the recipient who shows features of loss of microbiome homeostasis. And there have been a number of trials, whether in C. difficile infection patients, IBD patients, and there are most trials now emerging in conditions such as Alzheimer's or cardiovascular patients, for example, beyond the intestine. And what's already known is that this fecal microbiota transplant can not only restore microbiome homeostasis and the metabolite profile, but it can also restore intestinal barrier function. So what we have done in our study was that we were interested in looking at whether this transfer of fecal microbiota could actually have immunomodulatory effects or not. And this is a longitudinal study in which we had C. difficile infection patients receiving the FMT, and we have followed them on up to 4 weeks post-FMT transfer. And we've looked at a number of different markers of immune senescence. So firstly, confirming that post-FMT, we have seen a restoration of the microbiome homeostasis. We also saw a decline in your intestinal permeability. And we've looked at a wide array of immune markers. So we've got, for example, an expansion of your IL-10, which is anti-inflammatory. We see an increase in your naive T cells. But what was particularly interesting and grabbed our attention was that post-FMT, we start seeing a decline in the circulating senescent T cells, which wasn't something we were expecting. So how could changes in your microbiome, either they're causing possibly a relocation of these senescent T cells, or is it possible that we could have clearance or reversal of this senescence phenotype? So we thought the one thing to remember here is that this was all work done in C. difficile infection patients. So what we thought the interesting next steps possibly would be a FMT-based intervention in healthy older adults. And we were just heading down that pathway when the pandemic hit, and that put a pause on a lot of these plans. And that's what got us to rethink that possibly, rather than going for the transfer of the whole microbiome, we need to have a more refined and precision-based transfers to really dissect out what in that transfer is actually exerting this beneficial effect. So I know Caroline mentioned a little bit about the probiotics, prebiotics. The route that we are taking at the moment is more focused on the postbiotics to understand what are those metabolites that could be present. So we have a study ongoing now in which we are just transferring the fecal supernatants, rather the whole FMT, to see, and clinically it's shown promising evidence that it's enough to induce the beneficial effects of a whole FMT. And now what we are focused on is dissecting what is there in those supernatants, which are the metabolites that could be possibly having these anti-senescent features. So, of course, some of those key ones are the ones we heard a little bit about earlier on as well, such as your short-chain fatty acids. So propionate, butyrate, acetate, this is one of the key ones that have emerged, which possess immunomodulatory properties. So what we are doing at the moment is we've got a project focused on dissecting these interactions and to validate our findings before going into a clinical trial. So with the evidence, for example, for probiotics or prebiotics, the reason why we haven't always made that transition is there's still unclarity about which are the bacterial species of interest, how long do you need to have them for. So with our metabolites, what we are doing is we've got an in vitro model in which we can induce senescence in our T cells, and we are confirming whether they have any anti-SAPS phenotype. So we've identified a number of metabolites of interest that can show a downregulation of, for example, of inflammation of the SAPS secretion by T cells when in culture. We've also confirmed a downregulation of cellular senescence markers. And if you remember, I mentioned about some of those molecular mechanisms underlying senescence. One of the key ones was a buildup of DNA damage. So we've also confirmed with our metabolites, so this is looking at H2AX, which is a marker for DNA damage, that they can downregulate this accumulation of DNA damage, improve DNA repair mechanisms in culture. And another key mechanism on how they can work is that within aged T cells and our senescent T cells, we start seeing a built-up of dysfunctional mitochondria. And by targeting the mitochondria is one potential mechanism on how these microbiome-derived metabolites are working. So now that we've identified a few targets of interest, what we want to do is possibly validate them with animal studies, looking towards a future intervention trial using postbiotics or a synergistic approach in humans to combat T cell senescence accumulation and boost thymic output as well, possibly. So lastly, the other thing I want to discuss about, and this is carrying on from what Caroline mentioned, was about the diet. So if you remember in the previous picture I showed about how the metabolites, your diet is one of the key source of these metabolites. So your bacteria in the gut can convert these dietary fibers, for example, into metabolites of interest. And in our study, we were interested in dissecting whether differences in dietary patterns could also predict the trajectory, the immune aging trajectory that older adults are on. So it's well known that there are certain dietary patterns that can boost immunity. So we've heard about the Mediterranean diet, this high intake of fruits and vegetables, low consumption of red meats. But only 27% of the adults aged above 65 actually consume the recommendation of 5 fruits or vegetables a day. So to further integrate this analysis between your dietary patterns and the immune aging trajectory that you are on, we conducted a study in which we have recruited 40 young, 40 older adults, and we have done a number of screenings to access data on their diets. We have done a Mediterranean diet scoring, and in parallel, deep immune phenotypic functioning and analysis. And what we have done with support from our bioinformatician colleagues is a lot of data integration, and this has revealed some novel interactions between your diet and immune components. So one of the first ones that was particularly interesting is that if we've used our similar immune aging metrics, what you can see here is there weren't any significant associations in the young, but the red older adults, what you can see here is that those individuals who showed higher Mediterranean diet score were actually showing reduced immune age score. So there was a negative association between your Mediterranean diet score and the immune aging trajectory that they were on. And then what we wanted to do is possibly dissect out these interactions, because as I said, your immune age score is made up of 12 different immune components to see exactly which one could be possibly affected and what in your Mediterranean diet score could be having a beneficial effect. And just to give a little bit of a snapshot, one of the key ones that did come up is, for example, your dietary fiber intake here, which associated strongly with your immunological age, and more particularly this one with your senescent CD8 built up. The other key one that came up was this consumption of polyunsaturated fatty acids, and fish take was another interesting one that came up, suggesting novel links between our diet and immune patterns. So the other reason why this all links in together is that what we have observed that individuals who have there's an association between your dietary fiber intake and the levels of surrogate markers for your intestinal leakage. So by adhering to a diet which is rich in dietary fibers, you could actually be boosting your intestinal barrier function, and that might be an indirect way of contributing towards healthy immune aging. So what I would like to summarize here is that hopefully today I've convinced you all that there is a direct causal relationship between changes in our microbiome, such as loss of your intestinal barrier function, which is causing translocation of microbial products into circulation, which is driving immunosenescence and inflammageing. And the positive news there is that there are a number of targets that we have to help restore this barrier function and restore this microbiome homeostasis, which could be having a beneficial effect on the immune system. So I've touched upon possibly dietary-based interventions, such as adhering to a Mediterranean-rich diet or higher intake of dietary fibers. Fecal microbiota transplantation is another one. We are now working towards the therapeutic administration towards selected microbiome metabolites of interest. And of course, you have your probiotics and prebiotic supplements as well. So lastly, I would like to acknowledge members in the group and my funding agencies who have helped supported me to conduct all of this research, and thank you.